5 minuti dovrebbero essere tutti qua.
Okay, so please close the door. I'm going to start my class. So welcome everybody. Can you hear me from home, from people online?
Yes.
Yes. Okay, the volume. Okay, try to speak from home, please. Hello?
Yes, we are hearing.
It's very low, the volume. Can you try to speak, please, from home?
Can you hear me?
Is anyone connected, please, from the room and try to speak in the microphone so we can check the volume, please? If you speak in the microphone, because there's one guy from home and it's very low, I don't understand if it's a problem of my computer or of people from abroad.
Can you hear me?
Okay, yeah, I understood what's the problem is. It's the HDMI, so it's from here.
Yeah.
I need to switch, yes, this. Okay, so please try to speak.
Can you hear me now?
Yes, now it's okay.
Okay.
Thank you very much.
No problem.
Okay, the problem was the HDMI, so thank you very much. So welcome everybody. First of all, I would like everybody to switch off the microphone and just open it when you need to talk. Try do it from here. Okay, they're all off. Okay. Okay, so welcome again. First of all, I want to say that I'm very happy today because I'm going to talk and think about science instead of bureaucracy. So that's really an happy day for me. And I'm also very happy to welcome here in Bologna Professor Jaap Kandorp, is sitting here. Jaap is a professor from Amsterdam University. He will have his lecture this afternoon, 2:30, in presence here in the headquarters in Fano. And so this is basically the first slide of my presentation today. This presentation was usually made of 3 sections. The first one was more a modeling and mathematical development of population dynamics model. It was mainly on mathematical stuff. And then there was a second section on climate change effects, taking consideration especially the effect of temperature on population dynamics of model organisms that I'm studying and I'm specialized on, which are corals. And the third section, which is actually the only one I'm gonna tackle today, is about the effect of ocean acidification on population dynamics of corals. So basically, I cut the whole presentation, and I'm gonna only focus on ocean acidification to keep more focus and more straight on a specific goal, my presentation. So I guess you are aware of what is the meaning of population dynamics. The main goal of population dynamics is to understand the turnover rate of a biomass in a population. That's the final goal of population dynamics modeling. So we need to know the age of the population, the average age, the growth rate, the reproduction, and then after all this data, the goal is to understand how fast is the turnover rate of a given population, how long the biomass of a given population remain in a specific environment. I'm hearing there are people that are trying to get into the virtual room. I ask students, please, to check, to connect Teams and help me with admitting people in the class in order to avoid to stop the presentation. So I was saying I'm going to focus basically on ocean acidification effects on population dynamics, growth, and reproduction of corals. And I start with this slide that show you one of the 4 amazing sites known on planet where it's possible to study the effect of ocean acidification in the field. This is a volcanic crater, is a 12-meter depth out of the small island of Panarea in South Italy, in the northern Sicily, in the Aeolian archipelagos. And this is a quite unique place because, of course, all the Aeolian archipelagos, as you may know, is a volcanic archipelagos. But usually the volcanic activity and the vents are producing any kind of toxic compound, poisoning compounds, different kind of acidic substance like chlorhydric acid, sulfuric acid, and many other poisoning stuff. But in some place, for some reason that the geologists know very well, I'm not a geologist, I'm a marine ecologist, the system shift from a mixed system to a system that is releasing pure, like in this case, CO2, carbon dioxide. And there are 4 places known in the world that at a depth, at least, that are useful for studying the biology of photosynthetic organisms are known in the world. There is Panarea Island, where we are working. There is Ischia, where we are also working. And there is Papua New Guinea, near Australia, Papua New Guinea, where we are working with colleagues from Australian Institute of Marine Science. And then there is another place in Japan. So in Italy, we are very lucky for people that at least want to understand the effect of ocean acidification, because we have 2 of the 4 known places that can be considered as natural laboratory for the study of ocean acidification on the biology of organisms. Why? Because, as I said to you a minute ago, the vent in this case is releasing for the bottom pure CO2, pure carbon dioxide. So this means that in this place, the seawater is acidified by the reaction of the CO2 with the seawater, which is exactly the same mechanism by which climate change is acidifying the oceans. And in this specific place, as I will show you later, we have today the same pH conditions that are expected by the IPCC model for the ocean, for the global ocean, at the end of the century. So we are now able to study what are the adaptation, or better, I can say, the acclimatization response of marine organisms to ocean acidification, because today we have here the ocean of the future. So, as I said, I'm a marine ecologist, and I basically studying coral biology. And since the last 12 years, I'm more focused on the effect of climate change on coral biology in the tropics and in the Mediterranean Sea, because of the grant that we got together with people from Tel Aviv University, from Bar-Ilan University in Tel Aviv, Israel, when also Jeff Kendall was involved. We got this huge grant in 2009 this project ran for 6 years, from 2010 to 2016. It was a near sea. You see the logo here, was a near sea grant that we got together with Professor Zvi Dubinsky at Bar-Ilan University, Israel, with the campus in Tel Aviv. And because of this grant, we were able to study the effect of climate change on corals in the tropic and in the Mediterranean Sea, and we visited this specific place for many years, and we are still working on these sites nowadays. So basically, I want first of all to remind what is a coral. Of course, you are all aware of this word, coral, but coral is not a taxonomic word. It's a common name to identify a broad range of organisms. And the main characteristic of corals, of organisms identified with this word, is the production of an exoskeleton made of calcium carbonate. And corals usually are identified as organisms belonging to the phylum Cnidaria. And in the phylum Cnidaria, as you know, there are 3 main groups, 3 main classes. One is Hydrozoa, where there are few species, calcifying species, identified with this name coral, like a fire corals, like Millepora, that's the genus. Then there are Scyphozoa, the medusa, what we call medusa, the big medusa, that are not usually calcifying, there are planktonic organisms. And then there are corals in the class of Anthozoa. And when you find the word corals without any more adjective, without any more specification, we usually intend corals are organisms belonging to the order Scleractinia. Scleractinia. Actinia is the name that means anemone. Okay. Sclera means the skeletons. So Scleractinia are anemones, okay, polyps, that produce these exoskeletons made of calcium carbonate. So corals are basically Scleractinian corals. Usually we understand we mean Scleractinian corals. And that the word that I'm using here, corals, I actually intend, I mean Scleractinian corals. Corals so are polyps, basically. You know, in the phylum Cnidaria, we have 2 main phases in the life cycle, the medusa, the planktonic one, and the benthic one, which is the polyp. So corals are polyp, and so benthic, sessile organisms, which mean that they don't move. They live on the bottom, attached to a specific hard bottom, and they usually don't move from the place where they burn, where the larvae attach. So are sessile organisms, and we can divide the ecological mode of corals in two main groups according to the growth types and the energetic supplying types, the trophic strategies. So within the growth types, we can subdivide the corals in 2 groups, the solitary forms and the colonial forms. Solitary polyps, solitary corals, are individuals that are independent from each other. In this sense, we specifically intend the trophic independent, okay? And in this sense, we are also, in this room, the human are solitary organisms because we are not interconnected for a trophic system, the trophic modes. We are all independent one from each other. And the solitary organisms are independent polyps, as you see here, okay? And each polyps derive from a fertilized eggs. Okay, so usually the genotypes of these individuals are different. Okay, so then there is the colonial growth types you see here. The colonial growth is a cooperative way of sharing food mainly. Okay, so we have the first polyp, the founder polyp of the colony. In this model is the one is represented here in the center. So this is the founder of the colony. And then the other polyps that you see remain attached to the first one and to each other are produced by asexual reproduction. Without the involvement of sperm and eggs, they clone each other. Okay, so the first polyp, through clonation, form the other polyps, and so on in exponential growth. Okay, number of polyps, and they are all interconnected from a trophic point of view because the stomach that in Cnidaria is called coelenteron remain connected to the coelenteron of the neighboring polyps. So let's say that this polyp, the apical one, is catching some prey. Okay, the food that it catch is shared within the colony with the other members of the same colony. So it's a cooperative model. Okay. And as I said, the genotype of all these polyps in the same colony are identical because they are formed through a clonation mode of reproduction. Then we have the energetic supplying types, the trophic strategies within corals. We divide within this group the corals in what we call zooxanthellate and non-zooxanthellate or non-symbiotic. So let's start from the second one, the non-zooxanthellate. These are predators. Okay, all the cnidarians are predators of zooplankton. They prey their catch through poisoning cells that you may remind from your zoological study. They call nematocytes or cnidocytes. Okay, so they basically kill the zooplankton. The zooplankton are this member of these little animals living the water column that are transported by the currents. And when the planktonic organisms hit the polyp, these animals are killed by the cnidocytes. Okay, so the non-zooxanthellate behave as a predator, as let's say a normal animal. They are pure heterotrophic organisms. Then we have the zooxanthellate corals, okay, as you see here. And the zooxanthellate corals, you see the name zox. Zox is a common name that identify a group of microalgae in the phylum Dinoflagellate that in this case, they live in symbiosis with this microalgae. And this symbiosis is an intimate symbiosis because it's an endosymbiosis, which means that the dinoflagellates, the microalgae, living within the cytoplasm of the animal cells. So in the animal cells, in the cytoplasm of the animal cells, you find the nucleus, the ribosomes, the mitochondria, the other cellular organisms, plus the microalgae that living within the cytoplasm in special vacuole, and they are photosynthetic algae, and a great part of the photosynthetic, of the organic matrix that is produced by the photosynthesis, go into the metabolism of the animal cells. Up to the 90% of more of the energetic requirement of the animal cells. So basically, the zooxanthellate corals are also defined as mixotrophic, which means that their trophic system is a mixed mode of heterotrophy from predation and autotrophy from the photosynthesis. So they have a double type of energy getting into the metabolism of the holobionts. You heard this word from the first class, Marco Candela, okay? So the holobionts get the energy in two ways, from the predation and from the photosynthesis. In this non-zooxanthellate, non-symbiotic, is pure heterotrophic organism. So, as I start my lesson a few minutes ago, this is another pictures of the vents in Panarea, and where we study the effect of acidification in the field through the coral worm grant. You see here the name of Zydubinski, the name of people in Bologna involved in this project, and then Jep from Amsterdam was also involved. And the ERC provided a grant of 3.5 million, was 2009, and then we used this money for 6 years, from 2010 to 2016. And if you are not aware of the ERC, I suggest you to look at this acronyms here, take a notes, and please look at the website of ERC. ERC is the European Research Council. That's so can I finish my sentences? Then I take your question. Actually, thank you for getting into the speech. I did not remind you. Please stop me anytime. So the ERC means the European Research Council is the main founding body for basic research, or what they like to call it, frontiers research. So it's one of the few tools that we have in Europe for getting money from basic research. Most of the granting scheme from the European Commission are so-called top-down, which means that are applied research, basically. The European Commission wants to solve a problem and ask people to apply to solve a kind of problem, a given problem. In this case, the ERC is founding what we call bottom-up. Bottom-up. Any idea, any new idea coming from a scholar can be found through the ERC. So have a look at this website. Please. Can you please I ask you a question? If you can stand up and raise the volume of your voice so people can hear you from home. Thank you.
Just a curiosity, why don't you name it Vulcano Island? It's not a suitable site?
Yeah, there are a few studies there, also in Vulcano, but the vents are much more sulfuric. So there are few ocean acidification study coming from volcano vents, but still in the Aeolian archipelagos. But the amount of sulfur is much higher than here in the Panarea. Okay, other questions? Okay. So a brief introduction on climate change. You are, of course, well aware of this problem. You see, this is a report from the IPCC. The IPCC is the Intergovernmental Panel on Climate Change. It's a panel of scientists joined by different disciplines, of course. There are marine ecologists, geologists, geochemistry, like mathematician, modelers, whatever, like the strong science, but there are also scientists in the field of social science, policy, because as you know very well, climate change is affecting not only the biology of organisms, but is affecting the whole society, from the economy to the effects that this change are having in the deep ocean. So it's a very holistic approach. And this panel, as you know, is publishing every few years some report. And this is a report from this quite dated already, you see 2007. The last one was published last year. And if you look at the concentration here, you see this is in the time scale. You have a time scale of 10,000 years. Of course, the geologists, the geochemistry now are able to go back in time much longer than 10,000 years. But basically, the concept here is to see how the variation appear, but not in the range and in the rate of nowadays change. This is, for example, a graph from 2007. If I'm gonna update this graph with today's data, now we reach more than 400 ppm. So from 2017, where we were just above 350 ppm, now we are more than 400. So you will see this vertical line going out. Now we are here, out of the scale. Okay, so it's changing very, very fast. Okay, and as you know, the rate of change is the problem for the marine organism, for the organism in general, and the society as a whole, because the rate of change is faster, most probably, than adaptation rate. The rate of change is faster than the rate of adaptation of many organisms, and we use adaptation also in climate change science for the society. No? In climate change, we speak about mitigation and adaptation. Mitigation are all the policy that we may try to develop in order to reduce the emission of CO2. And adaptation are all the measures that the society can implement in order to protect their economy, their city, and their family. So basically, the problem is the climate change rate is faster than the adaptation, in any sense you want to use this word. If you want to use adaptation is an evolutionary word, no adaptation in evolutionary sense take times, take dozens, thousands of years, because need generation of many organisms and the natural selection to act. Okay. So basically, the change of rate of climate change is faster than any adaptation. That's the problem. And you see here in this little graph, the exponential increase from the second half of the 19th century, from the second industrial revolution, when the humans start using the carbon, the petrol to produce energy. And because of climate change, because of the CO2, carbon dioxide is also increasing the radiative forcing, okay? The force, the amount of heat that the system, the Earth system is accumulating. And because of this, you see the increase in global average temperature, of course, in global average sea level, and in decrease in snow cover and so on, things that you know and you are aware. In 2009, when we got this grant, we actually started working 2 years before, in 2007, when we met together, Steve Dubisky and other colleagues in Florida, in Fort Lauderdale, North Miami. It was an international congress in coral reefs, and we started thinking about the submission of this proposal. And the acronyms of this proposal, you see there, coral worm. We use the term worm. Why? Because at that time, the scientific community was very well aware of the problem of warming. Okay, but the problem of acidification was not still present. Okay, something that came a bit later, especially in a paper published in Science from our colleague, Jeff know him very well, Hove from Australia, with a very famous paper in Science, and that was 2009. So when we got this grant, the focus was warming, and the acidification was a little, there were few words in the original proposal about acidification, but then the work in Panarea became relevant in the next years because we became aware of the problem. The scientific community became more aware of the problem of ocean acidification. And this is 2009, 2010, so it's more or less a bit more of 10 years ago. So it's a novel, it's a current, a novel problem in the scientific community that is still going, that is still very deep investigated, because we don't understand many things yet on the effect of ocean acidification and on the possibility of organisms to acclimatize to ocean acidification. So there's a bunch of groups still working very actively in this project, in this problem. So here you see one of the many graphs that you can find on the website in different database papers. There are now there's a bunch of this data around, and these are data from the University of Hawaii. It's a marine station in the Pacific Ocean that show you the relationship between the concentration of carbon dioxide in the atmosphere that is increasing and the corresponding decreasing of ocean pH. Why the ocean pH is decreasing? Because the CO2, the more CO2 we put into the atmosphere from our activities, the higher become the partial pressure, the pressure to which the CO2 push into the ocean. So the more CO2 we put in the atmosphere, the higher is the pressure of CO2, and the more CO2 enter into the ocean. And as soon as the CO2 molecules touch the oceanic water, it reacts with the molecules, with the water molecule, you see here, and it produces an acid, which is the carbonic acid, which is a weak acid that immediately dissociates, okay, in bicarbonate, this ion, and the hydrogen ion, the protons. So the ocean is more and more increasing the concentration of hydrogen ions, and with the corresponding decreasing in pH. That is the way we measure, through the pH, we measure the acidity of any solution. So basically now there are predictions on how the ocean will change their pH during the next years. And usually the model by the IPCC are, at least until the few years ago, were at the end of the century. What will happen at the end of the century? Then scientists understood that the politicians that are the ones should implement the policy to decrease the CO2 emission are not very well, actually, are not at all interested of what will happen at the end of the century. Because the mandate of any politician, excluded the dictator, okay, are of a few years, 6 years, 5 years. It's a very short time, so the politicians have a very short time interest. Okay, so now the IPCC is also producing prediction at 2030, at 2050. They shorten their prediction to try to make the politician more sensible to the effect of climate change, with not a very well success, from what I understand, because even the last COP that was just finished a few weeks ago did not reach any substantial results. Please. Yeah, because so I hear you better, and also from home.
The question is, what is the correlation between the increase of CO2 amount in the atmosphere and the increase in pH? I assume that.
And the increase in the?
And the decrease in pH. I assume that the sea is somehow buffering the decrease in pH, but usually if you add, if you increase an acid and you have a buffer, at one point the buffer breaks, no? So my question is, if the correlation is, let's say 1-to-1, couldn't we reach one point where the buffer breaks when the pH decreases sharply?
So the buffer power of the ocean is less than the amount of hydrogen it is produced. That's the reason why the pH is changing. Now I'll show you later. The next slides, there is this famous cartoon from Hove that they explain the mechanism from a geochemic's point of view. So now you see here the prediction by the end of the century, okay? And it is the IPCC prediction are you see here. Today we have an average pH, ocean pH of 8.1, and it is expected that it will decrease down to 7.8 at the end of this century. So I'm also connecting to your question now. I'm sure you are you have experience working in the lab, maybe even at the high school, not only for your thesis at the master level. Working with buffer, the lab we use buffer from 8 to 3, so very large pH of many units. So we may wonder what is a decrease of just 0.3 units. But you have to remind that the pH is just the way we measure the concentration. And the pH is a logarithm transformation of the concentration. So if you apply the reverse equation, you transform the pH in actual number of hydrogen ions concentration, you may find out that 0.3 decrease in pH units correspond to an increase of the concentration of hydrogen ions of 100%. So again, in a very, very short time, marine organisms that are used to live in the ocean of a constant pH of around 8.2, 8.1 for many thousand, if not million of years, in few decades, they will find themselves living in a solution that is 100% more acidic than used to be. And this is in a very short time. So the problem is, are they able to adapt, or better, because we don't think they are able to adapt, to acclimatize, to change their physiology in order to cope with this big change in environmental chemistry. So there are many organisms that will be affected. The main one that we are focusing on are the calcifier organisms. We all follow a number of lessons during the last few days on calcification, calcifier organisms, and biomineralization. And we understood also yesterday from the lesson of Damir Kralj from Croatia and from people from Bologna on crystallization, and yesterday the lesson from Frédéric Marin from France, that there are different minerals that the natural selection select for building skeletons. I use this word now in a general way. Skeletons is any structure that is built, biologically built by an organism in order to sustain the body. That's the, from the zoology, a skeleton is an organ that make possible for an organism to sustain, to colonize the space. There are different minerals, but the most abundant minerals selected is calcium carbonate. We found calcifying organism in marine realm, but also the tree. In anywhere there are calcification, there are calcifying organisms. So calcium carbonate is the mineral that was selected mainly by the evolution, in the evolution for the formation of skeletons. And you see here different calcifying organisms. There are some of them living in the pelagic system, and we have some foraminifera, protozoa, they are calcifier. There are, of course, the coccolithophore, the microalgae, here you see, that are one of the main calcifiers on Earth. And then there are, of course, calcifier organisms in the metazoan, in the pluricellular organism, even in the plant system. Here you see in the pelagic domain, pteropods, these little mollusks, and then, of course, the mollusks phyla, there are many other important calcifiers, like the bivalves, the gastropods. And then there are calcifier organisms living in the benthic, at the bottom of the oceans. You here see a few examples of them, the echinoderms, like sea urchins, sea stars, holoturians. And then, of course, the corals. Corals, not only the photosynthetic corals, the zooxanthellate corals living in shallow water, like you see here, the reef corals, but also we know now that there are enormous coral reef in the dark ocean, in the very deep oceans, 1,000 meters depth, pure heterotrophic corals that are living in the dark environment, the deep water, so-called deep water corals, like the coral reefs of Lophelia. I'll show you later a few pictures. And also yesterday, Frederick showed you some estimation that basically remark or similar to the one I showed you today of the amount of calcium carbonate that is produced per year by all these calcifying organisms. It's in the order of gigatons, milliard of tons of calcium carbonate produced per year. And in this graph, you see the main calcifier. The forams, okay, are considered the main calcifiers, followed by the microalgae, the coccolithophores. Corals are also very important, echinoderms. Then there is a little part of calcification from the mollusk. And in total, the estimation is around 5-6 milliard tons of calcium carbonate per year. Milliard of tons. This looks a big number. A milliard of tons looks a big number. But try to translate a bit this number in infrastructures, in formation, in real things. Translates. Of course, you are very well aware of the tropical formation, like the GBR. Here you see these pictures, which is 1,000 of kilometers long along the east coast of Australia. And this formation, like the one also in the Maldives, the atolls, just to give you just some idea of the tropical formation, are providing something important. We are talking about ecosystem services now very much, no? They are providing a very important ecosystem services. Because as you know, as I introduced you, most of the corals are photosynthetic, at least in this environment, are symbiotic, zoox. So they are, all these things that you see here start basically from photosynthesis. Okay, so they, from the organication of matter from inorganic molecules. So they also are the beginner of the first chain of the food chain in the ocean. They start the food chain in the ocean that are usually oligotrophic water, but through the photosynthesis of corals, they start the food chain. And they sustain important economic sectors. There's no way that the most important economic sector in the world is tourism, for example. So that's an economic sector that this formation provide, sustain. Fishery, they protect the coastline from tsunami, from other impact from the ocean. But these things, these pictures are still coming from very far away from where we're sitting today. Okay, it looks like something far. But we can also look at what's happening just here, a few kilometers. If you swim and cross the sea, start in a few days, swim in a few days, you need to be a good swimmer. But let's take a little boat, just cross the Adriatic, go on the other side. In front of Ancona, there is a Mileto, southern Croatia, there is the Mileto Island, there is a coral reefs, a real coral reefs made of one of the endemic species of corals, of photosynthetic, zooxanthellate corals that we have in the Med, Cladochora cespitosa. Cladochora cespitosa was very well developed during the Pliocene, when the Mediterranean was a tropical sea. We had coral reefs of Cladochora all over, spread all over the Mediterranean Sea. Then, because of the climate change that happened during the Pleistocene and with the last glacial 20,000 years ago, most of the coral reefs of Cladochora were extinct, but there are still really coral reefs of Cladochora, not only in Mileto Island in Croatia. We have an important reef in Cyprus, and then some remaining in Calabria. There are also important reef formation in Ustica and so on. And then you have there at the bottom here, you have a Lophelia pertusa. Lophelia pertusa is deep water corals, and still the beginning, the study of the biology of this reef formation. This is an heterotrophic system. There's no photosynthesis down there. It's living thousands of meters of the deep ocean in the dark environment, and it forms reefs that are more or less comparable to the size of the coral reefs, the shallow water coral reefs, so huge formation, but at the bottom, in the dark part of the ocean. And then we have these things. These are the Dolomite in the North Italy, in the Alps. These are coral reefs, geological coral reefs. These were coral reefs. When the African plate hit the Eurasian plate, they emerge like a mountain. So that's the power, the significance of biomineralization. Okay, it's a really important process for our Earth, our problem, our planet. So, what's the problem of acidification? Acidification, you know, when you are at home, and for some reason the washing machine or any pipe at home is not working, what's the reason? Most of the time is because of the calcareous, calcium carbonate formation, are blocking the system. Okay? And what we put in the tube, in the machine, in order to clean the tube? Product. In Italy we have Mr. Muscolo. I don't know in England what you have. One is Mr. Then there is the other one was Mr. Muscolo, Mastrolindo. That's also different brand, different name, but the concept is the same. The one the grandmother were putting vinegar, thank you, or lemon. It's acidic. The acidic environment dissolves the calcium carbonate. Of course, in the Mastrolindo, the pH is not 7.7, it's much stronger, but that's the concept. The acidified environment attack the calcium carbonate. Okay, so as I'll show you later, and also you already understood from the previous lesson, that an acidifying ocean is making problems in two ways to the calcifying organisms. First of all, it may dissolve the skeletons already formed. Second, it can make more difficult for a calcifying organism to produce a new skeleton. Okay, so the acidification may attack in two ways: dissolving what is already formed or making more difficult to form a new one. Why? That's the famous paper of Hoth. This was published in 2007. Okay, so basically where we're writing the Coral War start, writing the grant. And this is a famous cartoon that was then reproduced in many different papers. But this is the way Hoth, in a very famous, a very highly cited paper on science, introduced into the scientific community for the first time in science the problem of acidification. That said, that's people, okay, warming, but we have another problem here. Okay, and through this very famous cartoon, it shows what I told you at the beginning of this lesson. We are producing CO2 as a med, like nothing, like a very high rate. You remember the vertical increase of CO2 concentration in the atmosphere. The more CO2 we put in the atmosphere, the higher become the partial pressure of CO2. The more CO2 go into the water, and when it go into the water, you see here, it reacts with acidic water, with the water molecules to produce carbonic acid that it dissolve. Okay, that is releasing two ions. One is the bicarbonate, and one is the hydrogen ions. The hydrogen ions is a positive ions, and as any positive things is looking for a negative. Okay, what is going to find in the ocean as a negative? At least two negative charge. The carbonate ions. See here, the carbonate ions. So this is the problem. So the hydrogen ions is going to react with the carbonate ions, okay, producing more bicarbonate. So basically, the higher is the CO2 concentration in the atmosphere, the higher is the production of carbonic acid, the lower is the pH, and the lower become the concentration of carbonate ions. And that's the problem. Because as you see, these are all equation equilibrium. And if I remove carbonate ions from the environment, the calcium carbonate that is already precipitated in the form of mineral tend to dissolve, okay, in order to reproduce the equilibrium of the system. So that's the way Hoth in this paper explain us the problem. Okay, the possible dissolution of calcium carbonate, of biologically produced of calcium carbonate by corals and other calcifying organisms that you see here. Okay, and with the problem for the ecosystem. Then there is another way to explain the problem through omega. Omega aragonite is the way the chemistry describes the saturation state of any solution. Okay, here, this is a poor chemistry. There's no biology in this equation. Okay, there's also a bit of physics because the product of solubility here that you see, it's also depending on temperature and on pressure, but I'm not going to the details of K today. I'm gonna put you the attention of the top of the equation, okay, where you find the concentration of calcium and the concentration of carbonate. So calcium can be considered infinitive in the ocean. Okay, there's really such amount of calcium in the ocean that is not actually a problem. The problem is the concentration of carbonate, okay, here, that, as we just seen, it decreasing with increasing the concentration of hydrogen ions. The more the environment is acidic, the lower is the concentration of carbonate. And if the concentration of carbonate tend to zero, omega tend towards zero value. And when omega is below 1, okay, the thermodynamics suggests that is favor the dissolution of minerals, okay, which means that the minerals that is formed, that is already there as a calcium carbonate, tend to dissolve when omega become low. When omega is high, instead, means that in the environment there's a bunch of calcium and carbonate, so they can react and they can precipitate, forming new calcium carbonate. So higher value of omega means precipitation, means formation of new minerals, okay? Lower value of omega means dissolution. And please take in mind this omega, because it will come out again this morning when I'm going to explain the biology of calcification in the experiment that we perform, because omega is also a measure of potential calcification, or is, let's say, can be considered a proxy of gross calcification, okay? Which means the facility, the probability of an organism to gross calcify, to produce, to start precipitation of new minerals. The higher is omega, the higher is the gross calcification, the potential calcification, okay? Which is different from the net calcification. That is another concept that is actually the mineral that is going to form the skeleton. So we have omega, which is a proxy of gross calcification. Okay, gross calcification is the potential calcification that is not equal to the net calcification, to the things, to the real minerals that we are going to find at the bottom of the ocean. Okay, well, this is the problem. Okay, we saw the problem from a chemistry point of view. The chemistry that is looking at this beaker in the lab. That's the way the chemist guy look at the problem. And then we saw more broad pictures, the ocean, okay, what's happening in the ocean. But the calcification, the growth of a skeleton is not happening in a beaker and is not happening in the open ocean. Where it is happening? Frederick explained you very well yesterday where it's happening, the calcification. In a, remember, confined what? Confined space. A very thin, confined space of a few micron, and not in the open ocean. So in a space where there is an epithelium of biological living things, cells, that they, through their physiology, they can modify this little space in order to maintain what you know very well, this concept and homeostasis. An environment that is optimal for calcification. The problem is the rate of change and the absolute change of the chemistry in the ocean make possible for these little cells to maintain the homeostasis. That's the problem that we are trying to understand. Today, we have some data, but we are still far away to make a final statement, if we will be able anytime in the future. So in fact, that's the anatomy of a polyp. Anthozoa polyp, coral polyp, that you find in any zoological book. Okay, a polyp is a very simple organism compared to the other metazoans. A polyp, like a medusa, a cnidarian, is made of two cell layers, basically. One is deriving from the endodermal cells of an embryo. When is this layer, we look at the embryo, we call it endoderm, but when it differentiates in the mature polyp, then we call it gastroderma, gastrodermis, which is the epithelial layer that is delimiting the stomach, or better to say gastrovascular, or how the zoology call it, coelenteron. The epithelium that is delimiting the coelenteron is made of gastrodermal cells. Then there is a non-cellular layer rich of protein, fibrils, water that we call it mesoglea, which is between the two layers, okay, here. And then we have the epidermis. The epidermis derived from the ectoderm of the embryo, okay, and it's delimiting the body of the organism, okay, and the epithelium that is at the aboral pole, which is the bottom pole here, the aboral pole of the polyp, we call it calicoblastic layer, calicoblastic, these little things, calicoblastic. What does it mean, calicoblastic? Plastic is a cell. Calico is again a word to indicate the calcification. Is the epithelium that calcifies, that is in charge of calcification. So I want to stress now, you see many times in my speech, here in the ocean is the climate change. Okay, here in this little space is the calcification. This is a very famous picture from Veron. Veron, a guy, a taxonomist of corals, very famous in our world, the coral world, from a book of Veron. We used these pictures in a book that I published with Zvi a few years ago to introduce this problem. And you see again how it is organized an anatomy of a polyp. You have a polyp, you see there is sitting, is literally sitting on its skeleton. Okay, and you see there with the green is the epidermis. Then you have with the white is the mesoglea, the red is the gastrodermis, and then there is a very thin layer that is even not colored here, which is here. Okay, down here there is a very thin, almost invisible layer of epidermis, which is the calicoblastic epidermis, that is the one in charge of producing the skeleton. Okay, again, here is the way where the ocean acidification occurs, and here in this little space is where the calcification occurs. This is a very like a famous paper from people Tamboutet et al. Journal Experimental Marine Biology. This group, our colleague Jeff is working with them, people from Monaco. It's the Monaco group. They're working at the famous Oceanographic Center in Monaco, in the Principate of Monaco. They are a very important physiologists in the field, and they show us with this infographic, an organization of a polyp. Okay, so again we have with the blue the ocean. Okay, where acidification take place. Then we have, this is the so-called oral pole, which means the pole of the polyps towards the sun, towards the water. Okay, you see this is the ectoderm at the oral pole, which is quite thick. Okay, where you find also the cnidocyte, the poisoning cells that the corals use to kill the prey. Okay. Then you have the mesoglea here, a cellular layer, and then you have the gastrodermis, okay, the gastroderm delimiting the coelenteron. You see, this is the gastrovascular cavity. the gastrodermis toward the sun, toward the oral pole, toward the sun, is the layer, the gastrodermal layer, is the layer hosting the ZOCs. The microalgae are mostly occurring within the cytoplasm of the gastrodermal cells, okay? And just a few words, because it's not part of this speech at all, the ZOCs are taken by the environment through phagocytosis, okay? So the gastrodermal cells form a phagocyte, a vacuole, okay? Within this vacuole, there is the living cells. There is a recognition of the immune system of the corals that do not proceed with the digestion, but maintain the living microalgae within this vacuole that it's become a symbiosome, okay? And the cells living within this vacuole where it does photosynthesis, and a great part of the organic matter go into the metabolism of the animal cells, okay? Then we have the coelenteron, and then we have the base, the aboral pole. At the aboral pole, you find, again, the gastroderms with no ZOCs, you see here in this model, the mesoglea, and then our actuals, the calicoblastic cells. Here, you see this thin layer of cells. These are the calicoblastic cells, and very important, with the orange here, you see the extracellular medium, the little micromicture space, the space in the thick of few microns, where the calcification take place. And then you see here the skeleton. So again, ocean acidification become in the open environment, okay, the calcification is happening in a very confined space, which is this one in orange. And then a few words on the physiology, okay? And on the biomineralization. First, let's see the biomineralization model. This is a paper from Couif and Dauphin from the Natural Science Museum of Paris, where they are based now. There's a paper in biogeoscience, and they explain the biomineralization model for corals. It starts from A at the bottom here, and it's finished at D. So you see here basic, in the model, they show 3 layers of skeleton already formed, okay, like a times 0, and the basal ectoderm, the calicoblastic cells. So it starts a production of new layers. What's the calicoblastic cells produce as a first things? What you learned yesterday, the organic matrix. This network of very complex molecules that are made of protein, but also glucid, also lipids, okay? And they are amphipathic molecules. So this means that have some part of the molecules are hydrophilic and other parts are hydrophobic. So they dispose the three-dimensional conformation according to the polarity, and within this organic matrix, there is the calcification that is governed by a physiological process that I show you in the next slide. So the organic matrix provide the framework within the calcium and the carbonate react to form the mineral. So the biomineral is in a great part formed by calcium carbonate, but there is up to 3% of the mass is also composed by this organic matrix. Okay, and then there is the role of magnesium. Again, I don't want to go in the details because already yesterday, Frederick showed you the role of magnesium, calcium in the formation of the different polymorph. You know already that calcium carbonate is one mineral, but can crystallize in different form, what the chemistry guys call polymorph. One of the form is aragonite, one of the form is calcite, and then there is a vaterite, and then there is also amorphous calcium carbonate. Okay, and there is the magnesium, as Frederick showed you yesterday, play a role in regulating this mineralization when the environment is rich in magnesium. Okay, from a thermodynamic point of view that Frederick explained you yesterday, the polymorph that is favored is aragonite. Okay, when the ocean, when the environment is rich in magnesium, the favorite polyform is aragonite. When the environment is poor in magnesium, the favorite polymorph is calcite. So that's the reason why they also put the magnesium here, because as this role in the formation of aragonite, that is the form, the polymorph that the current corals make their skeleton. So at the end, you have the new layer and so on. Okay. And now a few words on the physiology. The calicoblastic cells have an important role in regulating the chemistry environment, okay, of the medium where the calcification takes place to make this medium, this microenvironment optimal for the calcification, for the production of new calcium carbonate. So this is another paper from, actually this is from Cohen and McConaughey, that show the basic physiology. You see here the calicoblastic ectoderm in this model. This is a layer of skeleton already formed, and now this model explains how the calicoblastic cell modify this microenvironment in order to make it more suitable for the calcification. And it's very easy. You already know how they do it, because we understood already that the more acidic is the environment, the less is the concentration of carbonate. So in order to make calcification possible, you need to make the environment more acidic or more basic? More basic. So there is a calcium ATPase pump, okay, here you see, that has this goal, as the goal of removing protons from the water in this confined space in order to make the environment more basic, and in this way it increases the concentration of carbonate ions. Because you remember here, I go back 1 second, if I add hydrogen ions, I decrease the concentration of carbonate ions. If I remove the hydrogen ions, this equation go toward left, so it means that if I remove the hydrogen ions, I increase the concentration of carbonate ions. That is the one that I need in order to react with the calcium, okay? So what this pump does, removing proton, okay, at the expense of energy, ATP, and at the same time is increasing the concentration of calcium, which is not really relevant because the calcium is a lot, but that's the way the pump is working. So is increasing the concentration of calcium, removing proton, and is increasing the concentration of carbonate ions. So the two ions, the carbonate ions, the calcium, they can react and forming calcium carbonate. So the calcification is expensive from an energetic point of view. And the more acidic is the environment, the more proton you need to remove, and the more energy you need to spend. Okay, so that's the problem. Do they have enough energy to maintain the homeostasis of this microenvironment? So now we take a 5 minute break. Okay, if there are questions, I take it now. Otherwise, we take a 5 minute break before going on with the next section. Any question? Please.
To know if the corals are able to eat zooxanthellae in emergency case. So if they have not enough energy, if they can eat actively the zooxanthellae and the gastoderma or not.
Okay, thanks for the question. So now I'm working with a guy. If you want to put this name on your note, Todd de la Jeunesse, which is the real expert in the dynamics of this symbiosis, is the top scientist in the world specialized in this system. I can tell you that there is an hypothesis, a theory on how the corals recognize the zocs, because when the gastrodermal cells are the one I said to you are delimiting the gastrovascular. So their original job is to absorb the molecules after the external digestions. Because when they catch a prey, let's say that this is the Celenteron, the prey is inside. First of all, the gastrodermal cell produce enzyme that make an external digestion, and then they absorb through phagocytosis the food. Okay, with this mechanism they also absorb the zocs, the microalgae that are in the environment. Okay, they form this digestive vacuole that become a symbiosome. First of all, because there is a recognition of the zocs as hosts by the immune system of the coral. Second, there are a kind of experiment that show that until the zocs is vital and is producing it's vital, I mean, do photosynthesis, so provide energy, provide food, organic matter to the animal cells, the symbiosome remain as a symbiosome. When the zocs, for some reason, become sick or decrease the rate of photosynthesis or is not useful anymore, they digest the zocs. So in some case, yes, they can do it. Other question? Okay, good. So let's take a 5 minutes break. There was a coffee here. Yeah. Now, because I'm here, you did not provide it. So let's take a 5 minutes break. Thanks. Well, you see that. Io non li voglio. Non so chi l'ha portato, io no, non ho niente idea.
No, l'ho portato io per quello che volevo dire.
Per me è sempre complicato è complicato seguire queste lezioni.
Infatti sto cercando di essere più didascalico, cioè è interessante. Per esempio, io non studio la roba che fai te, però la roba che fai te a parte che qualcosa, diciamo, delle piccole connessioni ci sono.
Quando facciamo la parte di politica.
Esatto, però mi interessa molto.
Nella già trovata ce n'erano 36. Oggi quante ne sono fatte? Una in
Ma cosa? Ma per me possono fallire dopo.
immagini, oggi
C'è il Taralucci e il figlio del Taralucci e Pio. Allora mi devo pubblicare.
dice sì, no?
A un certo punto non è riuscito più. Mi sarei sentito bene.
Cioè, each errors of
non conviene iniziare perché non fa un inizio poi.
Stamattina. Ah, ok. Sono il tempo lo stesso. Sì, ci credo. No, non io.
Ma io ho questo di ascoltare.
Ad occhio, direi che
Però magari alla flash.
Ah, ok.
ieri
We arrivato.
Okay. So welcome back to the room. We restart. Please have a seat. Thank you. Thank you. Please have a seat. Okay. So, okay. So we introduced the problem of acidification, and we try, as I said you at the beginning, to afford this problem, to make some, to find some answer in the Panarea Island through a different approach. One was a short-term experiment, a manipulative experiment, when we transplant corals along the pH gradient created by the volcanoes, by the crater that I showed you before. So first thing, short-term experiment, transplanting corals along the pH gradient. The second one was describing actually the physiology and the acclimation potential of organisms, of calcifying organisms that are naturally living along this pH gradient. Okay, so there are calcifying organisms that are living naturally with their burn, they attach, they stay there for all their life along this gradient. So we tried the two approaches. So our model species were, of course, Mediterranean corals. One is Balanophilia europea. You see here on the left the pictures. Balanophilia europea is a solitary coral and is a photosynthetics or zooxanthellate or symbiotic. Okay, it does photosynthesis through its symbionts. Then the second species is again solitary coral, is Leptosamia pruvoti. Leptosamia pruvoti is a Balanophilia without the zoox, basically. Actually, many years ago, they were classified within the same taxon, then was divided. Leptosamia pruvoti was called sometime Balanophilia pruvoti. That was I want to stress this because basically it's the same animals. One does a photosynthesis, the other does not. That's the main difference. And then there is a third column, their third species, Astroides calicularis, is a colonial corals. So it's a different grow morph with respect to the other two. It's Astroides calicularis, also called in Italian Madrepora arancione. They are all beautiful, especially Astroides. It's very, very nice, also from a touristic point of view. They all belong to the same family, which means that they are very close relative. Okay. So basically, the difference between the three models are growth form and the trophic system. Okay, but they are all like the same organism. They are very close, phylogenetically close to each other. They are all belonging to the same family. So we can say the same animal with different trophic strategies or growth form. Okay. And this makes the comparison strong, because. So what we did. So basically, we were in Aeolian archipelago. You see here, Italy is the north of Sicily. There is this archipelago. Have you been there sometime? The Aeolian, some of the island. Okay. If you, for people that have not been there, please. Try the way to visit this amazing place. It's very interesting from the landscape, touristic, geological, naturalist, biological. It's very peculiar place, and it's a volcanic archipelago. You see, Panarea is this little island here in the northern part of the archipelago. Northern Panarea, there is Stromboli. Stromboli is considered the, or one of the, depends on the volcanologist, if it's the or one of the most active volcanoes in the world. Okay, it's very important for also the study of volcanology, and it's considered the or one of the most active volcanoes on Earth. And so all these archipelagos is very rich in any kind of emission, volcanic emission. And volcanic emission are rich of any kind of toxic compounds that are not useful for the scope of our research. I don't want to see if a coral or an algae or whatever you like, a fish, can acclimatize in sulfuric acid enriched environment. Okay. I want to know if it's able to acclimate in an acidified environment through the same system of the climate change. So I need CO2 to acidify the environment, not chlorhydric acid or other kind of acid. Okay, so there is one vent. This vent is in Panarea. In front of Panarea, there is a group of what they call archipelago of islands, are very small rock in the sea. Actually, there is one of the rock Dattilo that is big enough to host an amazing Roman from the Roman Empire villa, the beautiful mosaic. So at the top of this little island, this rich Roman guy built this house 1,000 years ago. Such a crazy, incredible, just thinking. This, and there is Bottaro. Bottaro is this very little island, rocks, okay? At the north part of Bottaro, here where there is the mouse now, there is a very strong CO2 emission. And in summer, you see the line of boat of tourists going to look at this bubble coming from the seafloor. It's quite amazing to see. But that bend that everybody knows is not the bend that is interesting for a biological point of view, because it's rich of sulfur and other toxic compounds. Very close, I would say 50 meters southern from the touristic CO2 emission, there is the far from the touristic emission, yes, there is the CO2 vents. This crater that from 12-meter depth is producing basically pure CO2. And the guy, I need to remind now this name, the guy that bring us for the first time in this place is Bruno Capazzoni, a geologist from the Department of Geology at that time, that unfortunately, 3 or 4 years ago passed away in a car accident. But Bruno was the guy that introduced us in these CO2 vents. Okay? He's the guy that discovered these vents. Okay, few years before we start working as a biologist, and he described for the first time the evolution of these vents until the time that it was, because this place started as a normal volcanic vents with all kind of toxic gas, and then moved to a CO2, pure CO2 vents. So in summer, you have these boats of tourists here looking at the main vents, and we were working actually 50 meters away from them. And we used to visit Panarea Island in summer, but also any season, in winter, in February, at least 4 times per year. And while in summer, in August, Panarea is full of people, and is one of the main touristic attraction, very expensive boat and the villa on the island. In summer is full of people. In winter there are 6 people living there, maybe 10, and one of them is the Carabinieri, and the other 2 were us. Okay, other 2, 3 were us. And the island people were calling us as the volcanologists, because they didn't realize that we were working on coral too difficult. We were the volcanologists. So it was very, very a great experience. Living there in the winter was really something. So what we did there, basically. So this is a short video that shows the approach. So you are all involved in marine ecology. So you recognize here a typical community, Mediterranean community, rich of Posidonia, and then you will see some fish around. Here we are 35 meters away from the center of the crater. And you see here the transplant. This is Astroides calicularis that is in this position because the colonies, they don't care about the light, Astroides. Okay, so you find Astroides in a natural environment, in the light environment, in the caves. They are heterotrophic, they colonize both habitat, light and no light. So we put it as we collected, okay. Then you see here Balanophilia europea. These are phototrophic, so they are obligated living in the light because they do photosynthesis through their symbionts. And then you see here the Leptosamia pruvoti. We put it in this position because Leptosamia pruvoti is pure heterotrophic, but in the natural environment, they colonize only the caves, the top of the caves, okay? They are not in a light environment. So we put it in this position to mimic the natural distribution. Of course, these polyps are glued with a non-toxic glue to ceramic tiles. So you see this, how we transplant, and I need to admit that this way to transplant was a failure. This experiment that I show you, this setting is not the one that produced the data, because this was the very first time we were trying the experiment, and we failed because the boxes were too high from the bottom. So in the winter, after the first storms, everything was completely destroyed. So we try many times before find a way to attach the corals in the position that we want, resisting the storms, the heavy storms of the winter, okay? So basically, this is the site one, okay? At the end, we found the way to fix the corals in the site one, and this is the site one where you see there is a normal Mediterranean community with Posidonia and so on. Now, the diver is swimming toward the crater that is down there. Okay, you see how the community suddenly, at some point, shift from a Posidonia-dominated community to a macroalgae, brown algae-dominated community. Okay, there's no Posidonia anymore. There are these algae. Some of them are even calcified, but most of these algae are not. Anyway, you see the change. And you see the crater, okay? So this is site number 2, which is closer to the crater, which means that it's already an acidified environment. And then you have the site number 3 here. This is number 3. This is one of us working in the field. And then you see here the number 4, which is very close to the center of the crater where there is the maximum emission at lower pH, the more acidic condition. So this is number 4, okay? Again, the number 4. You look at the column here. So this column is gas, is almost CO2 gas, almost pure CO2 gas. Of course, we checked with the geochemistry, we characterize in details of this gas emission. So this vent is producing a perfect pH gradient. There is a current from north to south. Okay, we almost never found an opposite current, maybe once, but the main currents come from north to south. So so from this direction in this direction, and this current is moving the gas, the bubbling, this direction, creating a perfect gradient. Perfect gradient, because think of the current. If some time goes this direction, then the next time goes in the other direction. Here it's variable. You will not have a very good like a gradient, okay? Because the water will always be mixed. Instead, there is this current, okay, coming from north, and this creating this perfect pH gradient. And you see here, this is the distance from the center of the crater. Okay, 0 is the center where the maximum CO2 emission, and then you have site 4, 3, 2, 1. Okay, and you see how the pH increase from the center, where it is the most acidic, to the periphery, where there is the lower, where there is the normal pH. Now, we have to understand how this pH match the prediction. You here have in the right the IPCC prediction for the end of the century. And so starting from site 1 you see has a pH of 8.1, which is actually the pH expected for the year 2000 to the ocean. It's a normal pH. Then site 2 has a pH of 7.9, okay, which is exactly the pH expected by the end of the century by one of the different IPCC scenarios. You know, the IPCC is providing different scenarios according to the possible different mitigation measures that the policy are taking to mitigate climate change, no? So site 2 is exactly matching the orange prediction of the IPCC. Site 3 with a pH of 7.7 is matching, that's the important aspect, matching the worst case scenario. According to the IPCC prediction, in the worst case scenario, which also is called business as usual, which means that we are going on to produce CO2 at the same rate as today, which is the most probable actually, considering what's happening at the policy, in the policy system. That's the worst case scenarios. In the worst case scenarios, the IPCC predict 7.7 as average pH for the next of the century, for the end of the century, which is exactly the pH that we have at site 3. And then we have site 4 with a pH of 7.4, which has a pH lower, more extreme than the pessimistic scenario. The worst case scenarios predict 7.7. Okay, in site 4 we have an extreme condition with a pH even lower than the worst case scenarios. Okay, so it can be considered site 4 as an extreme site. Okay, so what we did. So these are the raw data. This stuff was published in a paper a few years ago, of course, but I show you the raw data and not the modeling, the 3D modeling that we did with this data, which are, I guess, more straightforward from a didactic point of view. So we study basically the effect of acidification on mortality, tissue regeneration, calcification rate, reproduction of these 3 model species, okay? I'm going to show you today only one aspect because we don't have time to tackle all the problem, but it's the most significant maybe, probably is the calcification rate, the rate of calcification. So the question is if the calcification is in some way affected by the different acidification levels. So I'm going to explain you this table. So basically in each line you have the response of one of the species. In the first line you see the response of the photosynthetic symbiotic species, and in the other 2 lines you have the response of the pure heterotrophic animals, okay, the one that don't do photosynthesis at all. Then in each graph you have basically the calcification rate at the control site, normal pH, and at the 3 treatments, okay, at the 3 different pH. So you have the calcification at 8.1, 8.0, 7.8, 7.6, okay? Then the experiment was repeated in different seasons because we wanted also to understand if there was an interaction between temperature and acidification. Okay, so we did the experiment in winter. You see the temperature here and the months. In winter, in spring, let's go semi-warm or semi-cold water, okay, and in warm water, okay, with the higher temperature here. So what we got? We got, if you look at the first line, you see that independently from the season, from the temperature, you see that in the photosynthetic coral, there was no difference between the calcification rates at the control site in normal condition and in the acidified sites. See, look, this is the calcification in the normal site compared to the other one and so on. So in all the season, there was no significant difference or no correlation between the pH and calcification rate. If you look at the second line here, you see that's the solitary non-photosynthetic coral. You see that in cold and semi-warm or semi-cold season, the colder period, we have the same results. There's no relations between pH and calcification. But when in the warmer season, at the warmer temperature, we found a significant difference among the calcification rate and a relationship with the calcification and acidification. And this relationship indicates that the more acidic is the water, the lower is the pH. The more acidic is the water, the lower is the calcification rate. And also look at the y-axis here, because you have a positive and negative value, okay, of calcification. So this means that in warmer water, there is a relationship between level of acidity and calcification. With calcification, the decrease with increasing acidity. The last line, which is the colonial non-photosynthetic coral, you see that there is, in all the season, there is a significant difference in the calcification rate and a correlation between acidity and calcification, with calcification that decrease with increasing acidity. Okay. If you look carefully, the regression line, that model, you see that the regression line is intercepting the y-axis in one point here. And this point, this intercept point, is the point that is dividing the positive value of calcification with the negative value of calcification. See, in this season, the colder season, up to pH 7.9, the calcification tends to be positive. Below this pH value, the calcification tends to be negative, which means that there is dissolution, losing of skeleton, dissolution of skeleton, okay? If you look at the intercept point, again, you may infer a relationship between temperature and acidification. Because if you look at the intercept point, the intercept, okay, in the colder season is a 7.9, which means that below 7.9 start the dissolution. Okay, if you look at the intermediate temperature, you see how this point is moving toward higher pH. Okay, so it means that with higher temperature, the dissolution starts in less acidic environment. Okay, or if you want, the effect of acidification is stronger in higher temperature. And if you look even at the last experiment, you see how this intercept point is moving toward even higher pH, which means that there is an interaction between temperature and acidification in negatively affecting the calcification rate. Even in this line, there's the same results, because you see in the cold water, lower temperature, there is no effect of acidification. The acidification starts in higher temperature. Okay, and this is indicating that the effect of acidification is stronger with higher temperature. Okay, so these are the raw data that then were modeled, but the basic results here is that that there is at least one corals, which is the photosynthetic corals, that is not affected by acidification independently by the temperature. There's no effect in cold, middle, and high temperature. There's no effect. So what is the hypothesis that we, prego, please?
There's a couple of questions.
Please.
On the data. I was wondering, how long do you leave the samples on site before testing the effect of?
How long was the experiment?
Yes.
Thank you for the question, because this is the key point. Then?
And then, if during the year there are differences in the food supply in the water column, I suppose that in winter you have.
Okay, thank you for the both. First of all, the first one is the key point here, because is how long is the experiment, which is how long is the treatment. If you look at this at the date at the top of each column, you see that the experiment lasts 3-4 months. So the effect of acidification was maximum for 3-4 months. Okay, that's one point, and so we should not infer that the ZOX coral is not affected by acidification. This experiment means that in a short-term experiment, there's something in the ZOX coral that may help the ZOX coral, that it may help the ZOX coral to be more resistant. Okay. In a long-term experiment where the corals are acidified for all their life, we expect to see some effect on the population. Okay, but here the results, the take-up message is that there's something, there should be something in the ZOX coral that help him to be more resistant. Okay, that's the first thing. Second, of course, there is a difference in the season on the amount of nutrients in the water, but it's always there is no difference among sites. So the level of nutrient in the winter is higher than in the summer, but in both seasons is the same among the 4 sites. So the effect of acidity, there's no effect of nutrients among the sites. Okay, we measured that. As well as the temperature, there is no difference among temperature among the sites. I mean, in winter, the temperature is the same among the 4 sites, okay? As well as the nutrient level. In summer, the temperature is the same among the 4 sites, as well as the nutrients, okay? So the effect of acidification is not affected by different environmental conditions. Okay, it's the same. I answer your question?
Yes.
Okay. I don't hear you. The deep is the same. Yes, there's 1 meter difference. But that's not because the crater you saw in the video, no? There's a little slope, but it's go from 12 to 10, then again 11, go like this. Basically, it's the same depth.
Yes, I was wondering if in the case of Ectosamia cobati, yes, of course, we have the effect of temperature.
Yeah.
Since we see a correlation
an effect on the classification only in summer, maybe due both on temperature and both on lower supply of nutrients.
Yeah, but it's the same, the Balanophilia.
Yes, but with
Exactly.
the Samia is not so centralized.
It's not oxalate,
So
exactly.
it rely only on the
Only heterotrophy.
Yes.
Exactly.
So maybe both factor.
Could be, yes, the both factor, yes. But it's the same, also Balanophilia europea is in the same condition, but is a photosynthetic. In fact, that's the point.
Yes.
The difference is the same animal, in the same condition, one is doing photosynthesis, and the other is not doing. So there is less nutrient for both, higher temperature for both, and you have this difference reaction.
Very last things, just
Please.
curiosity. I noticed that the number of samples is different between the different seasons. Is this because some samples
Were missed.
Was lost or
Yes. Yes, there's a little variation. Yes. This is because of the storms, because we lost part of the data. Yeah. These are 3 experiments that we did. I think there are 6 or 7 more columns because we repeat. After the first 3 times, 2-3 times that was completely destroyed, then when we found the way to fix the experiment, we repeat. Okay, every season for 3-4 years. So this is just a part of the set. Yes.
Thank you very much.
Other? Yeah.
I ask you a comment more than
Please.
a question. I will comment that in the colder season, the maximum amount of calcification in all the 3 species was not observed at the lowest, at the highest pH. I mean, the maximum in every case was observed in a more acidic condition than the common.
In Astrale Calicularis, no. In the first 2, yes, you're right.
It seems also in the third. I mean, maybe I'm not seeing very good, but it seems that it's at 8 and not at 8.2. So in the third one.
Here, yes. Yes, it's in the 8.1, you have the higher value, but also you have higher value in the 7.6. So there's a quite variation. Yes, I agree. In fact, we're not significantly different. There's also the effect of manipulation. You need to take in consideration this. So the way we manipulate the corals all the same way. Okay, but some samples may be more affected by the manipulation than other. So there's also the manipulation effects that can affect this
So
growth
you
rate.
explain more with some observation error than a real pattern.
Yeah, there are this kind. You know, when you work in the field, you have this kind of problem. For example, it's hard to explain why these corals that is in 8.2 has such a low classification rate compared to this one.
So maybe
But that's the way we repeat the experiment several times, okay? And then we compare the data among, and then we merge the data. There's all the statistical. But it's hard to say to you why. I can say that I was not expecting to have such a low growth rate here compared to this one, but I also have to admit that the way we manipulate the corals could also affect, maybe this one was more affected by the manipulation that we perform on the transplant than, for example, this guy here. Okay, but in any case, you see there's a lot of variation, and at the end of the story, we found that there's no difference among this growth rate here. Here there was a difference among the sites, but there was no correlation. So the fact is that we never found any correlation in one species, and we found significant difference and correlation in other two species. And the work we did is the same. So the manipulative effect among the three model species is the same. But within the same species, within the same experiments, I cannot assure that the way the student glued this guy on the tail was the same delicate way that maybe another one of us glue the samples here. So you have also taken consideration this. But the way we perform the experiments, the level of manipulation with the error is the same in all the experiments and for all the different model species. So at the end of the story, we found this pattern that I show you here, and then if there are other questions, thank you.
Sorry, can I ask a question?
Please.
Maybe I miss the part you have explained this thing, but how did you measure the growth and classification?
In this case, was through buoyant weight.
Ah, okay. Thank you.
We actually measured the weight of the skeletons underwater through buoyant weight techniques. Yeah.
Thank you. Sorry.
Welcome, please.
Have you had any problem of predation, for example, from Sparisoma cadensis, since it's a Bremunde species, because I think it's new, individual in the environment, maybe some other organism can predate or, I don't know, destroy?
No, we did not recognize this. There are a lot of gastropods there along the vents. There are actually predators of these corals, but we also find predators, yes, on the tail sometimes, but we were not able to measure the level of this. This could be another.
Species.
Yeah. These little gastropods, there are bunch there actually, but we did not measure this aspect at all. Okay, so thank you for the. I try to move on because we also have the next part. Okay, so basically, what are the hypotheses that we made? First of all, one is on the photosynthetic activity. No, one is and the hypothesis is a kind of easy to formulate because, of course, when you acidify the environment, this acidified environment affects negatively the calcification to all the calcifying organisms. There's no calcification that is boost in an acidified environment. But, of course, if this hypercapnic environment, the rich CO2 environment, is acting on a photosynthetic organism, we may also suppose that more CO2 may fertilize the ZOCs. Not the single ZOCs, for example, because you know that the photosynthetic system at the end is saturated, the single photosynthetic, but the number of ZOCs that can be hosted. Okay, and in fact, I anticipate you. We are just working on this paper together with Todd de la Jeunesse, that we found an increase in the number of ZOCs in the corals living closer to the beds. Okay, not only the number of ZOCs, okay, so the same cells there are more ZOCs, but also the clade. There are different clades, different genotypes. The clades the acidic bands of ZOX are different by the clades that are living in the normal water. So there is an acclimation of the photosynthetic apparatus. So this hypothesis was made many years ago, right after the papers that was published on these experiments. Actually is in that paper, this hypothesis, and we are actually now proving that there is an acclimatization of the photosynthetic apparatus in terms of different clades and number of ZOX. Okay, so this photosynthesis could provide more energy to the corals that are calcifying. So the negative effect on calcification may be equalized by the positive effects on photosynthesis, giving more energy to calcification. So basically, the photosynthetic corals has negative effect, but also positive effect that at the end, the calcification remain constant. That's one first, then I leave you the words. That's the first hypothesis. The second one rely on the famous organic matrix. We did these experiments together with Giuseppe Falini, a colleague of the chemistry department, and was published already a few years ago, on the effect of the organic matrix on the capacity of calcification in a modified environment. So these are experiments in vitro. So what we did with Giuseppe, from different species, comprised the one are the players of these experiments, plus some tropical species. We extract the organic matrix from the skeletons of the different species. Then we put this organic matrix in artificial ocean, artificial seawater. We add calcium carbonate, and we modify the environment in terms of magnesium concentration. You remember that the magnesium is the one is affecting the polymorph, no? If there is magnesium, aragonite. If there is no magnesium, facilitated the calcite, okay? So we modify the environment, and what we found, again, this Balanophilia europea is really a peculiar coral in many ways. Many is always surprising us. The only corals that was able to the only organic matrix that was able to produce calcium carbonate in completely depleted environment of magnesium was the organic matrix of Balanophilia europea. All the other organic matrix produced calcite. The only one was Balanophilia europea. Again, what is the connection with the ocean acidification? This magnesium study, the discussion is that this organic matrix from Balanophilia europea looks like it's able to create a macro environment, okay, a crystallization environment, particularly independent by the environmental condition. At the point that in an environment where should not be produced aragonite, okay, is still producing aragonite. Okay, so this capacity of this organic matrix of producing a crystallization environment protected by the environmental condition could also help in the acidification environment. Third, this is the paper that we published just last year, I guess. It's the measure of the homeostatic capacity of this Balanophilia europea. This also was quite amazing. So I don't go into details, and I admit I'm even not a geochemist expertise, working with geochemistry. There is the boron isotope. Okay, we know that the concentration of boron isotope is geochemists know that the concentration of boron isotope in calcified structures is a proxy of the pH at which the carbonate structure was produced. So what we did in this Balanophilia europea along this gradient, we measure the boron isotope concentration, and that's the first graph. So you see some slope there, Okay, but in reality, then we measure that there's no statistical difference among the concentration of boron isotopes in the skeleton grew at different pH. Okay, what does it mean? This means that the pH at the calcification site, the pH at the site where the skeleton is formed in the medium, remember, the pH in this site, at the calcification site, is constant along the corals living along different gradients, or in different pH. Okay. See here, this is the pH at the calcification site, and this is the pH along the gradient, the oceanic pH. So you see the oceanic pH is vary from 8.1 to 7.7, and the pH at the calcification site is not different. So it means that these corals are very strong homeostatic power. And the last line is even more straightforward because it's the delta pH. It's the difference between the pH at the calcification site minus the pH in the ocean. So you see that how different become this pH. I mean, at the normal site, outside there is 8.1 and inside is 0.3 pH unit higher. So it's more basic inside and outside. And when the pH is decreasing in the ocean, the delta pH is increasing because, of course, if you have the inside pH that is constant and the outside pH that is decreasing, the delta pH, the difference is increasing. And you see how it can maintain a difference of up to 0.6 unit pH. So the pH inside can reach 0 unit higher than the outside in the ocean. And this is done by the activity of the calcium ATPase pump. Okay, so it has a strong homeostatic power. So there are other questions. What is the take message of this first experiment? Is the time. This is the short time experiments, which means that demonstrate that the photosynthetic corals, these species at least, are something that is helping compared to the non-photosynthetic, which does not mean at all that the photosynthetic coral in a long term, that it means for all its life is subjected to acidification, is not having an effect. Okay, it only means that as something that is helping. There are other questions? Please. Please.
Sorry. What is on average the replacement time of zooxanthellae within the corals?
This is hard to say. They have a generation time in the order of days. They reproduce very fast. Okay.
So it's able to change the clades within a few weeks?
That's not so easy. As there was someone talking about this, I don't remember who. No, forse Marco. What we know is that now it's unpublished papers, but it's going to be like submitted. What we know for sure is that the difference that there is between the clades hosted in the control site compared to the acidified, the distance, the genetic distance, is higher than the genetic distance of ZOX from Croatia compared to Panare Island. Okay, so what we know is that the clades the genotypes, okay, that are in the two acidified bands are very different from the genotypes hosted in the control side. And even the amount of cells is increasing. So there are more cells, more ZOCs, of different speech, of different genotypes.
Just in a few months, then we were able to change.
I don't know this. I don't know if it's. I guess it's something that they take from the environment, so that it's a horizontal transmission.
Do you take the same measure before starting the experiment?
No. We did this on the natural population that I show you now. Now, there's the last part is the effect of acidification on long-term effects. I mean, on the population of Balanophila europea that naturally living at these different sites. So now at the end, we were able to measure the photosynthetic rate, the number of ZOC, all the acclimation of the symbiotic system. But this, please. Okay, so let's take another 5 minutes and then I'm gonna do to the third section. Okay, thank you. Yeah. Questa è un'altra domanda importante. Dunque è esploso all'inizio del 2000. Quindi sono 20 anni, quindi non ci può essere un'adaptation, solamente per questo. Cioè, già l'adaptation secondo me è impossibile dal punto di vista teorico, perché mentre le zooxantelle hanno degeneration time di giorni, quindi possono anche adattarsi, cioè i dinoflagellati, i coralli hanno degeneration time decenni, anni, comunque. Quindi qua si parla di tempi 100 anni di climate change, come lo intendiamo noi. Quindi io l'adaptation nel corallo, nel compartimento corallo-animale non la concepisco teoricamente. Nel compartimento ZOX è possibile. Considerando l'olobionte nel suo insieme, tu devi pensare che c'è una parte animale, secondo me, che non si adatta, e una parte vegetale che invece potrebbe anche, teoricamente, avere degli adattamenti. Nel suo complesso, io lo vedo come una climatizzazione del sistema olobionte, e non escludo che facendo il cross transplant, per rispondere al tuo collega, cioè se io prendo dei coralli che vivono all'acido con quei clay, li trasplanto nel controllo, okay? Quindi il controllo li trasplanto nell'acido e poi vado a vedere dopo non so quanto tempo, anni, settimane, cazzo, se cambiano i clavi potrebbe anche succedere. Per quelli che vengono dal basico assumono i clavi acidi che li trovano nell'ambiente, quindi mostri anche una trasmissione orizzontale, non verticale dall'uomo, no? Oppure magari schiavi.
No, però è interessante, mi interessava.
Io secondo me cambiano come schiavi.
Mi interessava capire appunto se ci fosse già stato un adattamento oppure no. È interessante sapere.
Secondo me lungo quel gradiente lì è impossibile perché sta diventando. Quindi sono tutte robe di acclimatazione, non è poco, Cioè, un fenotipo vuol dire che lo stesso genotipo sostanzialmente produce fenotipi diversi, in grado di resistere ai cambiamenti ambientali, quindi magari favorire l'adattamento. Se non ti acclimati neanche.
No, beh, era solo curiosità perché in realtà c'entro poco in questo settore.
Sì, va.
Posso chiederle un paio di informazioni, diciamo, più burocratiche? Sarei interessato a dare un convegno a settembre e volevo sapere se avete già un'idea della settimana cioè delle date per la chiusura del terzo anno, no. Cioè non è un problema, io al massimo scrivo poi al massimo non vado.
Cioè tu che anno sei, scusami? Cioè al terzo?
Sì.
Sei 35esimo?
Sì.
Al terzo ci devi essere.
Ci devo. Cioè,
per passare l'anno ci devi essere. Sì.
Al
consiglio?
Sì, assolutamente.
Allora io ti dico, adesso non ho ancora guardato, onestamente non ce l'ho fatta. Cioè, su due balle, questa fotografia, veramente non scherzo, non lo so, però io tendo a copiare le date dell'anno precedente. Quindi se tu guardi il 30, cioè quando l'anno scorso io ho fatto il 34esimo, perché mi comporto così, no? Chi esce 35, allora diciamo li faccio per lui. Farò prima il 38, poi lo stesso giorno faccio il 37, poi faccio il 37, poi faccio il 35, cioè gli ultimi tengo. Quindi se tu guardi la data dell'anno scorso, quando ho fatto il 34, che sono quelli che uscirono, più o meno sarà la stessa data.
E nel caso secondo lei ci potrebbe essere l'opportunità di partecipare online o sarà obbligatorio in presenza?
Dovresti essere in presenza, devo essere onesto.
No. Perché comunque molti giorni magari posso andare sui primi due e poi...
Sì, ma non sarà proprio quel giorno lì, dai,
è che no, c'è 4 giorni.
Non so, dai, sarà proprio la settimana. Vai a vedere intanto quando ho fatto il 34esimo l'anno scorso, poi non ti regoli.
Posso chiederle o torno dopo, più che se vuole fare una pausa, perché ho altre 3 domande sempre burocratiche.
Ah, burocratiche.
Se no torno dopo.
No, dimmi, sono facili.
Io sono tornato venerdì dal periodo dell'estero. Mi ero segnato di dover mandare una relazione di fine periodo, però ho chiesto in giro, ho avuto pareri discordanti. Devo inviarla.
Pareri discordanti per farti pagare, cosa hai fatto? Marco Polo?
No, sì, ma è solo per da inviare una relazione a lei di quello che ho fatto.
Allora, pareri discordanti perché qualcuno aveva la Marco Polo e quindi la devi produrre.
E questa l'ho già fatta, l'ho già chiusa, questa pratica.
Avevi la Marco Polo?
Sì.
Ti hanno chiesto la relazione?
Sì.
Mandamela anche a me. Allora.
Va bene, perché è una relazione che ho mandato a gennaio, nonostante io abbia finito a fine febbraio, perché la Marco Polo mi prima i primi 3 mesi.
Ma mandami quella relazione. Va
bene.
Poi.
Se
mi serve qualcosa te lo chiedo.
Assolutamente. L'altra cosa è che sto provando a scrivere un articolo con il hobby della Spagna dove sono andato e loro sarebbero interessati a pubblicare in una rivista che dovrebbe avere un impact factor quest'anno, però non ce l'ha ancora. Io gli ho detto chiaramente che devo avere un impact factor, mi ha detto va bene, ne scegliamo un'altra, però vorrei appunto sapere se al momento della pubblicazione devo già avere l'impact factor o se entro l'anno. Io approfondisco
meno problemi che me, perché poi anch'io a me va bene così, però non è che decidi
no.
Decide il collegio dei docenti, di solito, però avrà un po' fatto. Non è che se no non ce l'ha.
No, perché penso che sia...
Perché a volte ci mettono 2 o 3 anni. Fai attenzione.
Penso che sia di 2 o 3, cioè è uscita da 2 o 3 anni, dovrei uscire quest'anno.
Fai attenzione, perché a volte ci mettono 2 o 3 anni.
Ok, grazie. L'ultimissima è l'anno scorso, per quelli del 34esimo, la scadenza della consegna della borsa della tesi era fine anno, è stata fine anno, giusto per un'idea.
La scadenza della borsa della tesi è agli uffici o al collegio?
Al collegio.
Il collegio è entro la data del settembre.
Ok, anche se c'è stata la proroga. Cioè, siccome c'è stata la proroga e è stato tutto programmato a gennaio.
Sì, quello il collegio noi teniamo fissi. Avrai più tempo dopo per mettere a posto tutti i dettagli.
Quindi a settembre vorrete già una porta.
Sì.
E giusto per avere un'idea, la porta rispetto alla versione finale deve essere praticamente identica? O...
Quella che manda il collegio dei docenti deve essere completa, se manca qualche pezzo. Quindi dovrebbe essere completa, forse manca un po' di roba. Cioè non dobbiamo valutare che tu hai fatto la roba. Se ci manca metà della tesi, possiamo valutare così. Se poi da quella porta che manca, quella che tra i reviewer, ci aggiungi qualche sezione che non va a stravolgere il lavoro che è supportato, va bene, perché è un festival.
Quindi i requisiti per gli articoli eccetera sono entro settembre o entro...
No, entro settembre.
Nonostante ci sia la prova.
Sì, la prova che ha cosa da poi per supportare la tesi. Per essere ammessi all'esame, noi quando facciamo il collegio di ammissione all'esame abbiamo detto che il paper, il periodo all'estero, la conferenza, la bozza della tesi e presentare il PowerPoint in presenza. E per l'ammissione all'esame finale, siamo noi che vi ammettiamo all'esame finale. Non è l'esame finale, cioè per essere ammesso all'esame finale devi avere il paper. Se tu sei ammesso, ma che fai la submission all'università della tesi, che vai al reviewer, che te la correggono, che metti a posto, che vai a fare l'esame. Grazie. Okay, so let's start with the last section. Grazie. Okay. Okay, so we understood that the transplant experiments was a short-term effects of acidification and relative responses. Now, as I said, we don't expect that on a long term, I mean, corals that are naturally living in acidified water for all their life, even if they are photosynthetic, they don't have any effects by acidification. But we want to prove this, no? So we also studied the effect on a long term on corals naturally living along this gradient. And we are also doing this now. Actually, we have just published the paper in Papua New Guinea, finding similar results to what I'm showing you today here in Panarea. The colleague is Katrina Fabricius. Want to sign this name, Katrina Fabricius, AIMS, Australian Institute of Marine Science, is the colleague that we're working in Papua New Guinea. So this is a paper published already quite ago, it was 2014, and on the effect of acidification on this calcifying organism. So along this gradient, there are several calcifying organisms. For example, you see there a gastropod, Dermetus triqueter. Then Balanophilia europea, the coral. And then there are other calcifying organisms like a microalgae. We saw the effect of acidification, Padina pavonica, for example, and other algae, other calcifying algae. So here you see the population density of Balanophilia europea and the mollusk along the gradient. You see in the y-axis the pH, yes, the pH at the four sides and the density of the organism. It's clear that there is an effect, I guess. Looking at the population density, you see there's a decreasing in the density of calcifying organisms until the point, which is site 4, where the population are not able to live. Okay. So that's the first data I want to show. So the Balanophilia europea is living along this gradient
with a decreasing in the population density until the point it completely disappeared at the site, at the most extreme site, that is with the pH 7.4. Then we measure the calcification, the net calcification. Which is the net calcification conceptually? Is the mineral that actually we found in the skeleton. And how we can calculate this calcification? From the bulk density, okay, and the linear extension rate. There is an equation, then I'll show you later, that relate the bulk density, the density of the material, okay, to the linear extension rate. Linear extension rate means how is the extension in the space, okay, it's millimeter per year, okay, that's the rate. Here you see the unit measure, okay, to the calcification rate, to the net calcification rate. So the net calcification rate conceptually is actually the amount of calcium carbonate that is deposited per unit area per year, okay? Which is different from the gross calcification rate, that is the potential calcification, that depends on the omega. Okay, there is a difference usually between the potential calcification and the net calcification. We will go back to this concept later. So look at the calcification rate. You see that there is a significant difference as well as in the skeletal bulk density. Okay, so if you look at the value here, there is a decrease, significant decrease from site 1 to site 3. Okay, 2.2, 1.9, and 1.8, as well as the same bulk density difference, significant difference we found among the sites. Here you see the authors list. JEP is part of the team of this analysis, as you see. And basically, if you want to make a graph of this relationship, you see the calcification, the net calcification rate decreasing, okay, with increasing acidity. At lower pH, there is a decrease in calcification rate. The linear extension rate, millimeter per year, which is how these skeletons occupy the space in the three-dimensional direction, okay, is constant. Constant is not significant different, okay. The bulk density decrease, okay, decrease with increasing acidity. The bulk density is the results, is the density of the material. So it's the results of the density of the material itself together with the porosity, the pores, the empty space. The more porous is a material, the lower is the bulk density, okay? So the bulk density depends on porosity. This porosity is a very important concept in and measure in this story. In fact, we see that the bulk density can decrease at lower pH because it sharply increased the porosity, which means that the skeletons in acidified water are more porous, like they suffer of osteoporosis. Okay, osteoporosis. They are weaker. And another fact that I want to stress, I will stress even later, is that while the size of the corals, because it depends on the linear extension rate, the size of the house is the same, okay, the density of the wall, of the material forming the house is lower because it is more porous. So it maintains the size, but the wall of the skeleton of the house become more porous, okay? So this is the question. Someone of you asked me, yesterday, Frederick, how can we estimate the classification rate, the actual classification rate? You can do it. You need to analyze the linear extension rate of your samples, of your organisms. You need to know how fast they grow, okay? And you need to know the density of the material. When you have these two factors, through this multiplication here, you can estimate the net classification rate, okay? So you see here, the net classification rate is equal the linear extension rate times the bulk density. So in acidified water, we have a decrease in net classification, okay? The linear extension rate remains constant. The skeletal bulk density decrease. Why? Because increase the porosity. What does it mean here in practical? I will repeat this 3 times, I guess, from just the third one, I guess. I've had the 3 times. What does it mean? Because that's important. Is after 1 year, after 10 years, whatever, the time lapse that you like, after a given time, the size of the corallites, the size of the house is the same, like in the control side, but the skeletal material is more porous, is more weak. That's the meaning, okay? And what is the biological significance of this strategy? Let's do this example. We are workers, okay? And we need to build an house. And in normal condition, they provide us a bag of cement. Okay? With all this cement, we build this house that I call normal house. Okay? This house has a space inside and a quite thick wall. Okay? That's our house. Let's say that we need to build an house, but with less cement. That it is actually what's happened in acidified condition. In acidified condition, the corals calcify less, so it has less calcium carbonate to build the house. Okay, so with less cement, and we need to build a house, we have at least 2 potential strategies that are, first one, I build a little house, a smaller house, with the same density of the wall, the same thickness of the wall. I have less material. How can I build a house of the same size with less material? Okay, so I would like to choose to do a smaller house, but a smaller house with the same thickness of the wall. Okay, it's clear, no? That's the first. The second choice is, no, I'm gonna build a house of the same size, but with a thinner wall. Okay, with less material, I cannot do, I have to choose, it's a trade-off. Or I maintain the density at the expense of the size, or I maintain the size at the expense of the density. I cannot do both. I have less calcium carbonate. So the corals decide to maintain the size. So what they do, same size, skeleton size, they maintain the size of the house, they maintain the size of the skeleton, but with a thinner wall, with a weaker wall. Okay? Why they decide to do this? Okay, why they do this? What is the gain and loss? You know that any biological organism must reproduce. That's the goal, the reproduction. But Paranophilia europea is reproducing only through sexual reproduction, to gametes. The corals need to do ovary and tests. Moreover, this coral is also brooder, so it is brooding, so it maintains its embryos within its body before releasing as a larvae. So this coral needs space for reproduction. That's the reason why it maintains the size, because it must go through sexual reproduction in order to reproduce. It does not clone. It's a species that doesn't know what is the clonality. It doesn't go through a sexual reproduction, okay? So it maintains the linear extension rate, and the gain is to have the space to host the gonads. The sexual maturity of any organism is not because of the age. They don't have the ID. It's because of the size. Organisms reproduce at a size at first sexual reproduction. They need to reach a size in order to host the gonads. And particularly, there are even some species that are brooders, so they even need to host the embryos. So maintaining constant the linear extension rate means that they can reach the size of sexual reproduction. And as you see here, this is another paper published a few years ago. You see the oocytes, the ovogenesis on the left and the spermatogenesis on the right. You see here the pH, the 3 sites, and the eggs. Okay, the young eggs, intermediate, and full developed eggs. Okay, you see how these have the same pictures at the 3 sites. So it means that even in the most acidic site, they reproduce. They're able to reproduce, to host the gonads. Okay, and this is the same for the spermatogenesis. You have here the 5 stages of reproduction and the reproductive stage of the tests, of the sperms. As you see here, the mature test, you see how they look the same at the 3 sites. That's the morphological analysis that demonstrates that the reproduction, the sexual reproduction is active even in the most acidic population. And this is the quantitative analysis. The size, frequency, distribution of eggs at the 3 different sites. You see how they are perfectly overlapping. The distribution of the eggs, of the oogenesis, and the spermatogenesis. You have here the 5-stage maturation of sperms, of tests, and you see as actually looks the same. That's the gain of maintaining the linear extension rate at the expense of skeletal density, because they are obligatory sexually reproducible. Okay. What is the loss? Of course, the loss is the weakness of the skeleton. Okay, the skeleton are more porous, so they are more fragile. They can be more easily attacked by predators, by endolytic organisms, and so on. In fact, we see how the population density decrease with the decreasing of the density of the skeleton. Weaker skeletons, weaker populations. Less density, less population density. But when I tell these stories to my students, I always want to point out a positive aspect of this story. If you look at this, this is a prediction for 200, before the end of the century. The green line is the latitude of Panarea, where the experiments and the studies was done. And you see how the oceanic pH is changing, okay? This is at the pre-industrial, before the second industrial revolution. Okay, it was 8.2. Now in 100 years, we already decreased to 8.1. And then you have the different model prediction by the end of the century. Okay. So look at the worst case scenario, 7.7. Okay. What the population of Balanophilia europea does at 7.7 is here, is living as a weaker skeleton, but is sexually reproducing. When the population of Balanophilia europea is disappearing at 7.4, which is not predicted by the end of the century. So this is a good point. Show us how there is an acclimatization potential of this and maybe other corals, because we are finding the same results even in Papua New Guinea. Okay, so these corals at pH 7.7, which is the worst case scenarios, is living and is reproductive active. So the loss is the mechanical strength of the skeletons, but the gain is the sexual reproduction. And if the corals acclimatize, means that they can reproduce. And reproducing means the production of a new generation. The production of a new generation through sexual reproduction means genetic variability. And genetic variability means the possibility of adaptation. Okay? So that's the hope that comes out from this experiment, from this study, okay? Now, the last 3 slides, 4, I don't know, are the more difficult. Could be difficult, but I'm sure we will succeed as usual. So you remember the omega concept, okay? The omega aragonite. We said, one thing is the net calcification. The one I can measure, because it's the multiplication of the skeletal density that I can measure by the linear extension rate that I can measure. So there's a real things. Then there is the gross calcification, potential calcification that depends on omega. Okay, that is the chemistry environment. Okay, so look at this omega here. We saw before the boron isotope indicate that the pH is the same at the different sites. Remember the boron isotope story, the homeostatic. So if the pH is the same, the calcification site, the pH of the calcification site is the same, I expect that the omega is the same. Why? Because omega depends on the pH, right? If the pH decreases, we have more acidic water, the carbonate ions is going to disappear and the omega go towards 0. But if the pH is constant, the carbonate ions concentration is constant, right? So with constant pH, I expect omega, constant omega. And it's in fact is exactly what we measure. Okay, constant, no difference, significant difference. If the omega is homogeneous among the sites, we expect a potential calcification homogeneous, right? The gross calcification. And in fact, is exactly what we found here. You see this up in this graph is the gross calcification at the different sites, and you see how there's no significant difference. But the net calcification is decreasing. We just saw it. It's half an hour that we speaking about the decrease in net calcification, right? The net calcification is decreasing and the gross calcification is constant. How is it possible? The potential calcium carbonate that is precipitating in theory is the same, but the one is actually found in the scoral skeleton is less. What does it mean? That part of the calcium carbonate is disappearing. There is another equation, a second one, this one, that relate the net calcification to the gross calcification and the dissolution. Remember, we start saying that one of the problems of acidification is dissolution, no? Okay. So the net calcification is equal the gross calcification minus the dissolution rate. So let's say that the gross calcification is 5, a number, okay? The dissolution rate, there's no dissolution, is 0. How much is the value of net calcification? 5. Okay, there's no tranello in this questions. Okay, now I'm gonna have some questions, but there's no tranello. It's obvious. Okay, but if the dissolution rate is higher than 1, than 0, okay, is a positive value, so the net calcification is as a lower value compared to the gross calcification. So in our case, we have the net calcification that is as a lower value compared to the gross calcification. So this means that there may be or there is a dissolution rate. So part of the calcium carbonate at the start is not going to build the skeleton, but is going to be dissolved in the acidic environment. And the very last slide, then I'm done actually, then I have the conclusion, is this one. This is a model. We published this model, which is also a theory actually, last year, I guess. No, it was 2 years ago already. So this is summarizing all these stories. And I'm going to explain what does it mean. Focusing on the porosity. At the end, we will understand how the porosity could be formed. What is the origin of this porosity? Okay, so let's start from the blue sector. You see here, this is the site 1, the normal, and the site 3, okay, the acidified condition. You see here the pH in the seawater, 8.07 and 7.74. So you have a higher pH here, in the site 1 compared to site 3. Higher pH means higher omega. Okay, in fact, you see the omega is 4, basically. If you take out the decimal, is 4, and here the omega is 2. Okay, so this is more saturated environment, a more basic environment. So you have then you have the calicoblastic cell. Okay, in the calicoblastic cell, we have the calcium ATPase. Okay, that the role of the calcium ATPase is one to maintain optimum condition the chemistry of the calcifying fluid. Okay, so you see here that we measure the boron isotope stuff that we talked this morning, the pH and the omega at the calcifying fluid does not change. They don't change, okay, between the acidified ocean and the normal ocean. What does it mean? This means that the calcium ATPase pump must work stronger in the acidified water in order to maintain the same omega, the same pH. Right? Because in this medium here, enter water from the ocean, because here we have in this medium calcifying fluid, the fluid is the water also, the water comes from the ocean. So the water coming from the ocean is more acidified. So these pumps need to work more in order to remove the protons and maintain the optimal pH, right? In fact, you see here how the work in this model of the ATPase pump is double, okay? Is higher respect to the work that the pump ATPase do in the normal condition. Then you have the third concept of the gross calcification, okay? Because omega is constant, okay? The gross calcification is constant, but we found that the net calcification, the actual skeleton, is decreasing. The net calcification is less than the gross calcification in the acidified environment. Why? Because it's increasing the porosity. Okay, the more acidified, the net calcification is lower because the density of the skeleton decreased, because increased the porosity. What is the origin of these pores? Is random. It depends only on the aggression of the seawater, of the acidified water that they dissolve part of the skeleton. How these pores are formed? That's the big question. And now we try with this theory to answer this question, and I want to try to answer with you. So let's make a joke. Okay? Let's say that one calicoblastic cells, in order to maintain the optimum pH at the calcification site, in the normal chemistry environment, needs 1 joule of energy. To maintain the optimum pH needs 1 joule. Let's say that in the acidified environment, to maintain the same pH needs 2 joules. Okay? Let's say that the energy in the environment, the energy that these cells can use is the same in the two places. Let's say that is 10 joules. Okay, 10 joules and 10 joules. But here in the normal pH, one cells need 1 joule, and here one cell need 2 joules. Now the question for you is, how many cells can be activated? How many cells can work in the normal pH compared to the acidified pH? Let's say from the normal pH, how many cells are activated? 10. In the lower pH, 5. So let's imagine a carpet of calicoblastic cells, okay, made of 10 cells, okay, a layer. In the normal pH, they are all working, right? You said this, they are all working, so they form a layer of skeleton. In the acidified pH, you said how many cells are working? 5. So you have 1 work, 1 not. 1 work, 1 not. 1 work, 1 not. What they form? What is the results of yes, not, yes, not, yes, not? What is the results? The pores. Where they work, they form the skeleton. The cell that is not working is a pore, right? So what we suggest, this theory, and we're working that is published stuff, is that the porosity is biologically controlled. And it depends on the amount of energy that the cells require in order to maintain the chemistry constant. Okay? There's not enough energy. Even if the amount of cells, we know now, the amount of ZOX is increasing, the clade of ZOX is changing, but the amount of energy that you need to calcify, to have optimal calcification at 7.4 is higher than the system can provide you. So you form a porosity, you form a pores. So we expect that the effect of acidification in non-ZOX corals should be much higher because they don't have any help from the ZOX. So at the end of this story, that's the last slide, what we did during the last 15 years and more. First of all, we studied the effect of temperature along a latitudinal gradient. I didn't touch this argument at all today, so I give you just the main results. We compare always the same corals, ZOX, not ZOX, from Genova down to Pantelleria. It's 1,000 kilometers, 2 degree increasing average temperature, seawater temperature from the north to south. We found that the ZOX corals are affected by increasing temperature, negatively affected by increasing temperature, while the non-ZOX corals don't have any effect. This is, of course, a long-term study, okay, because we were studying natural population. So then we have the short-term experiment, the transplant experiments, comparing all with ZOX and non-ZOX, okay, the one I talked to you this morning. And we saw the ZOX corals having something helping in the acidified environment compared to the non-ZOX coral. That's the result, the results of the short-term experiment. The ZOX coral have something helping compared to the non-ZOX in the acidified environment. And then we have the long-term exposure, the CO2, the one just finished now, the long-term CO2 study on the ZOX corals, where we found the negative effect on the population because the population is decreasing, the calcification rate is decreasing, but there's also positive data, no? I stressed it before that even in the most acidic condition, the population thrives, is surviving as a weakened skeleton, but is still reproducing and is still alive. What we are missing in this story is this question mark here, is the effect of acidification on non-ZOX corals on a long term, okay, on a long-term scale. And now in Panarea, we found an acidified cave, still not have published nothing. We are now producing the first thesis where we are actually studying with the same methods that we did in Panarea, the effect of acidification on solitary corals, so it's the same system, same growth form, living in a cave from the entrance, which is the normal condition, down to the cave, the tip of the cave where there is the more acidified environment. And we are now producing the first data, and I can't wait actually to update this presentation with this data that we have now. What is the take message of this story is that, of course, there's much work to do. That's quite straightforward, but we have some signals of potential acclimatization of these corals that, of course, need to be need more studies. Okay, that's what I have for you. If you have any questions, we are done. It's 12:40, so we have time for having lunch and also for a bit of discussion if you want to say something. Also from home,
Just curiosity.
Yeah.
Before you mentioned the experiment in Papua New Guinea.
Papua New Guinea.
And you say that you are observing the same results. But,
Yeah.
I assume that the species you are testing are different.
Yeah, different species, same results.
So,
On the skeletal scale, the porosity, the density.
So I was just wondering if the species you are testing in Papua New Guinea have the same characteristic of
They are all photosynthetic, yes.
And about the sexual reproduction?
They are colonial corals.
Colonial, so.
Yeah, so they can, they do both. The difference, in fact, thank you very much. When you're comparing stuff, you need to compare, you need very take in consideration the growth form. So a colonial coral is not relying obligatory on sexual reproduction. So it can have different like strategies. Okay, most of the species there we test. I think we study 6 different species, and the acidified water, in most cases, we observe this increase in porosity. Okay, but we don't have the data on linear extension rate and age. We only have the data on the density. Okay, so we saw an increase in porosity. Okay, so which is a decrease in skeletal density, similar to Balanophilia europea, but we're missing the linear extension rate. So we still don't have all this part. Instead, in Arianna is there, right? Is Arianna? Arianna, together with Jep and other students, are working in these cave stories of these solitary corals, and which is Caryophylla ornata. Caryophylla ornata, we study the reproduction of these corals all over the many stations in the Mediterranean, and we found that these corals most probably rely also in asexual reproduction. So it's not sexually obligatory. Okay, it's not sexually obligated. So, and what we found in the cave, we found that these corals is decreasing the size with acidic, in the acidic water, the corallite are smaller. Okay, and the density of the skeleton are the same or even higher than the control side. So this solitary coral compared to Balanophilia is supporting the hypothesis that the sexuality, the reproductive mode is play a role in the strategy. Because in that case, it's not sexually obligatory. You rely also on sexual reproduction. And we found the opposite pattern. Small corallites with very high density in the acidic environment. Welcome. Other question? Please.
For me, it was quite wondering that for reproduction, in particular, you did some histology, no, of gonads. That there is no difference in the different condition of pH. But have you already tried to look for other modification, for example, in the contents of the eggs? Because
the fecundity is the same. At the polyp level is the same.
And the quality of the eggs, then the energy budget used for the egg, not the size. For example, the molecules inside the egg. I know that.
So the deutoplasm look the same, but your question is quite interesting. I can tell you another story if you want. No, but go on.
Yeah, no, because I did some work on fish population reproduction.
And what did you find?
No, not my studies, unluckily, but yes, there is a different content in mucins or other molecules related with the quality and then the size of the embryos in particular. Coming from the egg in different condition of pH, but also seed temperature.
Okay, so I have two stories to tell you. First of all, is in Panarea. What we found, we published this stuff last year in Knowledge Oceanography. You find all this paper in the web Marine Science Group, or I can send one. So what we found is that the fecundity, the number of eggs is the same. Okay, so but because of the population density is quite less than in the control site, you have the number of embryos produced per unit area is much lower. Okay, that's because it depends on the population density. Okay, but if you do the ratio between the recruits, the number of primary polyps, and the number of eggs, okay, there is an order of magnitude of difference between the two sites. Means that to have one recruits in the acidic site, you need 10 or 100 more eggs or embryos than at the control sites. I mean, in order to have one recruit in the acidic site, you need much more larvae. So this is an effect probably of a larval mortality. So you have a much higher larval mortality in the acidified site than in the control site. That's something we quantified this. Then in this study here, in the long term, along this is not acidification, it's the temperature gradient. We quantify the gametogenesis in terms of number of sperms and eggs produced in each of the population. We studied 6 populations, as I told you, from Genova down to Pantelleria. So there is a irradiance and temperature gradient, okay? So that was the scheme. So in Balanophilia europea, we found that what we define as a reproductive efficiency decrease with increasing temperature. Means that in order to have one mature egg, you need to start for an order of magnitude number higher of little eggs. So there is a kind of mortality of eggs. Okay, at the coldest sites, the number of smaller oocytes and the number of eggs is the same. There is a ratio of 1:1. From one little oocyte, you have one egg, one fertilized egg. In the more warmer side, the southern side, warmer side, in order to have one egg, fertilized egg, you need to start for 450. Okay, I show you this this stuff quite.
The reproductive effort is higher, much higher.
The reproductive efficiency is lower because you need more small oocyte in order to have the same number of fertilized eggs. Okay. In the non-ZOX coral, there's no difference. There is a ratio 1:1 along all the gradient. If you have 1 second, I'll show you this data. Here I am. Here.
So this is a story that we published few years ago already. So you have, for example, no, not for example, on the left you have Leptosamia pruvoti, which is the non-ZOX, and the right, the Balanophilia europea, which is the ZOX. You see the temperature gradient, that's the model, okay, that's the final model that we published from the raw data, etc. So you see the temperature, the range temperature is the same because they were actually studied exactly in the same sites. They were collected, the two species at the same time in the same sites. It's exactly the same population. So you have the same range temperature. You see, this is the population density of Leptosamia pruvoti, which is not different. From Pantelleria up to Genoa, they have like a homogeneous population density of 3 kilograms of skeletal biomass per square meters, or a number of individuals is here, around 10,000 individuals per square meters. Okay, and this is the population density of Balanophilia europea, which is significantly decreased from lower temperature to higher temperature. So the question was if this decreasing in population density was caused by a decrease in the reproductive activity. Okay, so here you have here summarized the results for Balanophilia europea. This is the fecundity. The black line is the number of mature eggs along the population is the same. Okay, an average sexual mature polyp has 250 mature eggs. Okay, that's the number. Okay, and this instead is the number of small oocytes, young oocytes that are produced in the same population. So this means basically that in the colder site at 18 degree average temperature, the number of immature oocyte, the one started developing, okay, is the same like the mature one. There's a ratio 1:1, is 100% efficiency, okay. At average way between Pantelleria, between Genoa and Pantelleria, average temperature, middle temperature, you see how the number of small oocyte is higher compared to the number of mature oocyte. So you need more little oocyte in order to reach the same number of eggs, and even worse in the warmer population. Okay. In contrast, in Leptosamia pruvoti, the number of young oocytes and mature oocytes is the same along all the temperature gradient with no effect of temperature. Even in this along the temperature gradient, then that's it. We found that in southern population, in warmer population, the calcification decrease. Okay. Okay, like in the Panarea, and the strategy that the corals use is the same. It maintains the skeletal, the linear extension rate, even in this case. So the temperature has the same effect of acidification on the calcification of this coral, and the strategy is the same, maintaining the size. Okay, there are other. Please.
Maybe a late question, but the porosity, the pores that create, are they open to the environment?
Yeah.
So the question is, could the increased pores allow the installation of biofilms or interaction with other, I don't know, microorganisms or other species?
So the more porous is the skeleton, the higher is the surface exchange, of course, with the environment. So that's one for sure. Now we are, as Marco showed you the first day, I'm not sure if there is a relationship between the porosity and the microfield, the microbiota, for example. I cannot say, but I'm saying that the microbiota, the bacteria, the population of bacteria are changing along this gradient and make the metabolism more heterotrophic. More autotrophic. The nitrogen fixation is increasing. The bacteria that fix the nitrogen are much more important as a population in the more acidified side than in the control side, making the metabolism more autotrophic. But I cannot say that there is a relationship between the population of bacteria and the porosity. I don't know, I never think about that. For sure, increase the exchange with the environment, the number of pores, because increase the surface connected with the environment. Instead of a solid, no, if you make pores connected with the seawater, with the environment, it increases the exchange with the environment. I don't know if it can help with the respiration. I don't know, would be also. Welcome. Okay, so thank you very much. Let's have a snack, and we see you here at 1:30. No, 2:30. Yes, thank you very much. Okay, thank you also from people, for people from abroad. I'll see you next time. Goodbye.